right?
Thanks.
Okay, so my name is Krista Peltoniemi, and I work as a senior scientist at Luke. And I've been working with microbes or microbial ecological stuff about over 20 years now. So it seems like a long time, but there is always new things coming. So there's never like every day is different. But now I'm going to present to you my results from my two projects. So the one has now ended, but the other one is going on still a bit further. And the other one has been funded by EU Horizon program. But the first, so the main theme of these results are the organic farming and how it affects on microbial diversity and communities. I tried to change the slide, but it doesn't, at least for me.
Yeah, same for us.
Okay, let's see, nothing happens. What's the problem here?
Should you stop sharing or what? And try again.
Okay, now
No,
at least
yeah.
it's changed.
Okay.
Let's hope it works. Okay, this is just a background image about the theme. So there's many articles and results that show that the organic systems are beneficial to microbes, and it has a great impact on the microbiomes or carbon and nitrogen content itself, and total phospholipid fatty acids or enzyme activities compared to conventional farming. And it has been also shown that higher microbial diversity in organic farm fields, and it has many advantages or positive things to the soil. For example, to carbon sequestration, greenhouse gas mitigation, and nutrient and water uptake, and pathogen control, etc. So we have studied this organic farming in Toholampi, which maybe you can see the small spot in this Finnish map over here, and this is the image from the sky above the field plot. So it's a long-term experimental field, which has originally started in the 2001 to investigate the impact of nutrient leaching. And what is specific in this experiment, that it had two different farm types included. So it had cereal farms and milk farms, which produces the like dairy crops producing field. And they both had organic and conventional farming in these plots. And now in this, you can see what kind of a cropping cycle they had. So it had the 4-year different crops in this cycle. And our samples or investigations were made in this second cropping cycle year. So there was barley in this conventional cereal farm plot, and then the other plots had Timothy clover or ley, or meadow fescue with ley crops. So we had samples in 2018 from the first 20 centimeters of bulk soil, and we had 2 samples in this same year. So we had samples in spring before sowing, and then autumn after the harvest. And then we investigated different things. So microbial biomass, carbon and nitrogen were investigated with fumigation extraction, and then microbial activity with CO2 produced by gas chromatograph, and then microbial amounts with qPCR, and lastly microbial community composition with sequences and coordination analysis with Permanova. So in the next slide, I will show you some results. So in the first figure, you will see the microbial CO2 production, and it's clear that this organic farming in cereal farms had a higher activity compared to conventional cereal farms, but there isn't any, and that happened in autumn samples. There was a slight increase also in spring, but that wasn't a significant one. And then the rest of the images are about the copy amounts. So the middle, you will see the fungal ITS copy amounts, and you see the same pattern as in this activity figure. So there is more fungal ITS copies in organic cereal farms compared to conventional cereal farms, but there isn't any difference in other farm types and within productions. But there isn't any impact seen in these bacterial gene copy amounts. And next slide, there is a microbiomass, carbon and nitrogen, and very similar patterns as you already saw in the previous images. But there is also a change in spring when you look at the cereal plots. So there is an increase in organic cereal plots compared to conventional one. And there are some changes in this dairy crop production, but they were hardly significant, or they were actually not significant at all. Of course, there is a lot of variation, but this was a clear pattern shown in these cereal plots. And when we look at the community composition for bacteria and fungi, they are here. This is maybe a bit complicated figure, but these different colors represent the different farm types as well as the production types. And there was a change due to the farm and due to the system both, but the bigger change was due to the system for fungi. So it explained like over 20% of the variation in fungal community in autumn, and less in spring, even though these all were significant changes. And the farm type explained even more, so whether there was cereal or milk crop. So it explained greatest in autumn for fungal variation. And for bacteria, it was a bit less significant. Oh, sorry. Okay, so we go into this other project, this EU project called Soldiver Agro, and just as a background, what is it? So this is one of the work packages we wanted to kind of investigate what is the basic status of soil biodiversity under organic and conventional farms. To be more specific, they were all wheat fields around Europe. So we had almost 200 samples or sampled fields from 9 European different pedoclimatic zones under organic and conventional farming. And so we had samples from Finland to very southern Spain and everything between. So we had samples in 2019 after the harvest in every zones, and we got first 25 centimeters of bulk soil from each of these sites. And we have done lots of analysis, including all all kind of chemical and physical soil analysis, for example, bulk density, soil pH, nutrients, aggregate sizes, and so on. And from some of these sites, we have already also measured greenhouse gases, but not from all. And then microbial communities were investigated as in the previous project with from Illumina and Amplicon sequenced reads. So here are the results from alpha diversity measures. And I have to say that I haven't done this bacterial data, so I just kind of borrowed this image from my colleagues, so I cannot tell much about their work, but this is just to show that there isn't so much difference between bacterial richness between different management types in any of these regions or zones. But when you look at the fungal diversity images in this right-hand side, there was some differences in some zones. For example, in Emerald zone, which is actually Estonia, and then also in the southwestern zone, Lusitania. So there were more fungi in organic management in these sites. And also, if you look at the B image, there was a similar pattern in this diversity index, which was Shannon in this case. Then we did some models from these richness indices, and there was clear that the fungal richness was bigger in conditions with higher long-term precipitation, and also with those with lower temperature within last 12 months before sampling. And also those sites which had lower carbonate content and lower available phosphorus amounts. So there was a clear correlation with these conditions with fungal richness. But when we looked at the community composition for bacteria and fungi, the pattern was kind of wasn't so clear. So there is hardly any significance between these organic or conventional systems, but the differences between these zones or regions, they're very large, and that make the most of the difference. So in this fungal ordination, there's a clear north-south change. So the communities were very different in north compared to south, according to this analysis. And when we look at the fungal groups, these kind of a functional or ecological groups, we could see that there is some effects to some symbiotrophs, which are these important fungi which are taking care of nutrient water chains between crops and soil. So there is also, you can see also that there are more symbiotrophs, or relative abundance of symbiotrophs are larger in this Lusitanian zone, which is this southwestern Spain. And also we saw some significance in continental zone as well. And also the same pattern lies in pathotrophes in this same southwestern Lusitanian region. So I go into the conclusions. What we can say based on this Boreal study, long-term study, that this farming system affects microbial biomass as well as fungal amounts and both bacterial and fungal community composition. But the impact of crop plant is also very large, especially for the fungi. And well, I didn't show you all the details, but we also concluded that the absence of manual fertilization and more frequent tillage practices likely explain the divergent results for conventional cereal crop fields, which was shown in these results. And the European scale results showed that this organic farming generally increased fungi, but the diversity increased only in some regions in overall. And it was clear that the fungal diversity itself is affected by local weather and soil conditions. So whether the cooler and moisture there is, or whether there is less phosphorus, and whether the soil is more acidic, there is clearly more fungi. And we also could see that there is more symbiotrophs and beneficial fungi, although their amounts were quite small in this data, but there is a difference. And also more pathetrophs in organic farming at least in northwestern Spain. And that might be because of the lack of fungicides, but there might be also some other reasons, of course. But here are the articles which are you can go and have a look at more detailed results and more results if you are interested. And that's about it. And I thank you for listening, and I'm ready to answer your questions. And here are some figures from my dearest colleagues and the Soil Development Group in the meeting last summer in Estonia. Thanks.
Thank you very much. Interesting topic. Is there any questions? Stefan, please.
Hello, thank you very much for your talk. So very interesting. I have a question about, so you show that you have an increase in diversity and amount of microorganisms, and you also show that the proportion that is affected between beneficial and pathogen is depending on the area. But at the end, in this organic soil, do we see more resistant plants? Is there any incidence on the protection or on the resistance of the plants? Do you have some data on this? Yeah, if there is any beneficial effect at the end on the crops.
Yeah, unfortunately, we didn't look at the plant health in this data at all. So I don't have that kind of information, but of course that would be the very important point in this case. But I don't think we have data from that perspective.
Yeah, I think it will be very interesting. Yeah, because you showed all the actually. My first question was about actually beneficial versus pathogenic, but at the end of your talk, you gave some data.
Yeah.
But then at the end, do we see some actually results on the protection or the resistance of the plants? Yes. Thank you very much.
Yeah. Thanks.
Yes, thank you very much for your talk. I was very interested. Do you know what about the soil carbon? Do you have more soil carbon in the organic system or not? And what about the nitrogen use efficiency of the plant system or the system? Do you know something about that?
Yeah, there is another article from the soil characteristics, and I'm not sure whether it's published. It's probably not yet, but it's coming. So I cannot really, because I didn't investigate that so much. So I don't know about. I cannot really answer that, but it's coming. the data is there and it should be published in the near future, so you can go to have a look at it. So there is plenty of articles coming from this project still.
Thank you.
So is the project over or close
No,
to
it
be?
actually ends in the end of May this year. So there is
Okay.
like a final works going on and articles in preparation now. So
Thank you.
Is there already possible to give some advice to the bio or the organic agricultors? I mean,
yes, there is attempts to try to look at something, but of course it's a bit challenging because we all know that even though there is some general rules how to farm organically in EU level, there is still many differences how it's done in different parts of the Europe. So there might be some like general different patterns. How do you farm organically in Finland, for example, compared to Spain? So it will be very hard to say those, to tell or kind of find these general patterns for all the Europe, but maybe at regional level that might be possible. But of course, maybe we don't have enough, like we should have more data to build this larger views to say something so comprehensive. But that was of course our aim initially, yeah.